Good morning, and my name is LaDonna Wyatt, and I am a junior biology major at Morgan State University. Today, I'll be presenting the impact of Nanofit 25S zero-valent nanoparticles on growth and localization in the genetically engineered Cyanobacterium diplocyphum. The objective of this study was to identify the effect of iron nanoparticles on growth and total lipid content in F. diplocyphum. About 85% of our energy needs are met by fossil fuels such as coal and oil. The extensive use of coal and petroleum-derived fossil fuel has resulted in climate change, pollution, and other adverse environmental effects. As the need for a more cost-effective fossil fuel increases, the demand for new sources of biofuel is amplified. The use of biofuel will reduce about 50 to 70% of carbon dioxide emissions. Cyanobacteria are photosynthetic prokaryotes that have been recognized as a potential source for biofuel production. Cyanobacteria are valuable for their wide range of applications in both the pharmaceutical and the cosmetic industries. They're an ideal organism for this study due to their fast generation time, minimal nutrient requirements, and high net energy converting biomass into fuel. Diplocyphum strain B481SD is a model cyanobacterium and exhibits impressive response to stress factors, making it an ideal candidate for biofuel production. B481SD was chosen for this study due to its enhanced lipid production and complementary chromatic adaptation, which allows the organism to adjust their pigment content in response to changes in light. The figure to your left is a figure of diplocyphum grown on a BG11 agar plate. In recent years, nanoparticles have gained invested interest in the biodiesel industry due to their high surface area, reactivity, and light scattering properties. These nanomaterials are known to enhance biohydrogen, biogas, and even bioethanol production, thus increasing energy conversion efficiency. There are a wide variety of nanoparticles, but for this study, Nanofit 25S zero-valent nanoparticles were chosen for their ability to enhance cellular growth and metabolic activity in photosynthetic microorganisms. The figure at the bottom of your screen is a figure that shows B481SD cultures grown in the presence of nanoparticles. This study aims at determining how nanoparticles affect the growth of B481SD cultures for biofuel production. In addition to determining how B481SD cultures exposed to nanoparticles can be visualized. To identify the distribution and the localization of nanoparticles into cyanobacteria cultures, field emission scanning electron microscopy and an energy dispersive spectrometry was used. Diplocyphum strain B481SD cultures were grown in BG11 media and exposed to Nanofit 25S zero-valent nanoparticles at concentration 0.4, 0.8, 1.6, and 3.2 milligrams per liter. Cultures were grown under constant shaking at 28 degrees Celsius and 70 RPM. Cultures grown in media containing 0.5 milligrams per liter of FeEDTA served as the control. 3 replicated treatments were maintained, and growth at optical density 750 was measured at 72-hour intervals. Data from the study was analyzed using ANOVA and Tukey onyx significance testing and reported as cumulative treatment means. Results are shown in Figure 3 for the growth of F. diplocyphum strain B481SD cultures over a 15-day period. Looking at Figure 3, we can see that on day 6 was the first day where cultures that were grown in the presence of nanoparticles showed significant increase in growth over the control cultures which were grown in the absence of nanoparticles. This trend continued until day 15. On day 15, final significance was measured, and it was shown that all cultures grown in the presence of nanoparticles at concentrations 0.8, 1.6, and 3.2 milligrams per liter showed significant increase in growth over the control cultures, which were grown in the absence of nanoparticles. B481SD cultures were grown in BG11 agar media and exposed to Nanofit 25S nanoparticles in liquid BG11 media at concentrations 0.8, 1.6, and 3.2 milligrams per liter. Diplocyphum cultures were grown until day 12, and energy dispersive spectrometry and scanning electron microscopy was performed to validate the localization and the distribution of nanoparticles. Field emission scanning electron microscopy is a microscope that uses electrons instead of light, and energy dispersive spectrometry is a method used for elemental analysis. Both were used in this study to validate the distribution and the localization of nanoparticles. Results revealed that the percentage of iron in the EDS spectrum for B481SD cultures grown in 3.2 milligrams per liter of nanofer 25S zero-valent nanoparticles was 6.75%, while that of cultures that were grown in the presence of nanoparticles at concentration 0.8 milligrams per liter was only 0.73%. Cultures exposed to 3.2 milligrams per liter of nanofer 25S zero-valent nanoparticles exhibited the highest percentage of iron in the EDS spectrum, while control cultures showed no iron on the EDS spectrum. We use elemental analysis by atomic percentages as a semi-quantitative method to correlate the nanoparticle concentrations in the control and the treatments. Looking at figure 4A, we see cultures that were grown in the absence of nanoparticles. Here we see no iron indicated on the EDS spectrum. Looking at figure 4B, we can see cultures that were grown in the presence of nanoparticles at concentration 0.8 milligrams per liter. Looking at these results, we can see that iron was present at the low quantity of 0.73% for cultures treated with 0.8 milligrams per liter of nanoparticles. Then when we compare that to figure 4C, which shows cultures grown in the presence of nanoparticles at concentration 1.6 milligrams per liter, we see a slightly higher quantity of iron at 1.85% on the EDS spectrum. Then when we look at figure 4D, which shows cultures grown in the presence of iron nanoparticles, we see the highest concentration of iron on the EDS spectrum at 6.75%. And finally, figure 4E shows iron nanoparticles alone for comparison. While the application of nanoparticles in green technologies can be toxic to microorganisms, at low concentrations, they can promote the growth of reactive oxygen species. At non-lethal levels, nanoparticles can cause oxidative stress, which is known to increase lipid yield in cyanobacterial cells. Oxidative stress induced by these nanoparticles could have augmented lipid production. In accordance with previous studies, total lipid yield of 26.2 to 35% was observed in algae Tridiscus minutus when cultivated with 5.1% milligrams per liter of nanoparticles. In conclusion, cultures treated with 0.8 milligrams per liter, 1.6 milligrams per liter, and 3.2 milligrams per liter of nanofer 25S zero-valent nanoparticles exhibited significant increase in growth when compared to the control. Using FESEM and EDS analysis, it was shown that cultures exposed to 3.2 milligrams per liter of nanoparticles exhibited the highest percentage of iron, while no iron peak was observed in the spectrum of the control cultures. Future studies are aimed at identifying total lipid yield and polar lipids in nanoparticle-treated B481SD. In addition to maximizing biodiesel potential of F-diplosyphon for efficient large-scale biofuel production. In future studies, American Standard Method will be performed. We also hope to develop new clean energy sources. The following slide shows all of my references. I would like to express my sincere gratitude to my advisor, Dr. V.G. Sither, and all of my collaborators and lab members. In addition, I would like to thank the National Science Foundation for funding my research. Thank you.